Okay, good morning class. Once again we are here to discuss asexual reproduction, and today we are going to talk about three topics under asexual reproduction. We'll talk about fission, we'll talk about budding, and we'll talk about spore formation. So when we classify asexual reproduction, we get a few channels we'll discuss. Let us start with fission. Fission is of two types. Number one, A, binary fission, and number B is multiple fission. As the name suggests, binary fission and multiple fission. Here the fission is going to occur once, that is single, and here the fission will be multiple times. So first we are going to discuss binary fission technique, and the example would be amoeba. The example will be amoeba. We will take a diagram. Suppose this is the amoeba. The next procedure will be, it will form a plasmodium. It will form a curved structure. The nucleus will divide into, the nucleus will elongate here. The nucleus has elongated, and then the third is division of the cytoplasmic material and the nucleus. Okay, first we see the there was the parent cell. This is the parent cell. It has elongated, the nucleus is elongated, and then it has divided into two parts. The cytoplasmic, along with the cytoplasmic material and the nucleus. It is divided into two parts. And now these are the offsprings, or we also call it as daughter cells. The fission has taken place once, and due to single one-time fission, the parent cell has got divided into two, and these both will grow individually into a new individual. Now let us talk about multiple fission. In case of multiple fission, what's going to occur? In case of multiple fission, beta, this point happens several times. It will divide multiple of times. This whole process, this both binary fission and multiple fission, these both take place only in case of unicellular organism. This process takes place in case of unicellular organism. So let us concentrate over multiple fission. In this case, there is an organism. There is an organism, a single cell. It has a nucleus. So in the first process, an additional protective covering will be formed over the cell called crest. And this protective covering is so hard that it cannot be broken down by any means. I mean to say, any means, in normal conditions it cannot be broken down even by hammers. Normal hammers can't break it, though if we apply tremendous pressure we can break it. In case this crest will be formed, this is a protective layer, and inside the crest, this nucleus will start to divide several times. Now what does it look like? This is the crest, this is the cell, and the nucleus has divided number of times. And after this, each nucleus will bind with a small cytoplasmic material with it, and we see a number of young cells or the daughter we see here only 2 daughter cells are formed, and here many daughter cells have been formed. In comparison of these two techniques, multiple fission is a faster technique, as because in this case at the same time there is a spelling mistake. At the same time 2 daughter cells were produced, but in this case more than 2 cells or daughter cells are produced. And one big difference is this binary fission takes place in favorable conditions. For any process to take place in any organism, it always requires favorable conditions. We all require favorable conditions to do our activities. So this reproduction technique, binary fission, happens in favorable condition, and this multiple fission technique takes place in unfavorable conditions. This takes place in unfavorable conditions. Since the conditions were unfavorable, that was why crust was formed over the cell so as to protect the cell from any type of breakage, any type of damage which may be caused due to different environmental factors. And it stops after division of the nucleus and cytoplasm. This cell remains at this position until favorable conditions arrive. Means this crust has been formed, cell has been divided, nucleus and cytoplasm has been divided, but these daughter cells will not be released unless favorable conditions arrive. Favorable conditions may include favorable temperature, favorable humidity, pH, etc. And as soon as the favorable conditions come, these cells, daughter cells, will be released, and a lot number of young cells will be ready to grow. And the example for this will be Plasmodium. Example for multiple fission is Plasmodium. Example for this is amoeba. This process has taken place once. This has taken place multiple times. There is formation of crust in case of multiple fission, no crust in binary fission. This takes place in favorable condition, and this takes place in non-favorable conditions, unfavorable condition. Now we will discuss the third point, that is spore formation. So our next topic is budding. Example for budding is yeast. This is a very simple process. Yeast, suppose this is a yeast cell, a bud will start to form, another bud, and similarly one or more than one bud may be formed depending upon organism. This is yeast. If we take an example of Hydra, in case of Hydra generally one bud is formed. If this condition arrives, this is called chain of buds, and this condition we call it simply bud, because there is one bud only and here there is a chain of buds. So in case of budding, when favorable conditions be there, यहाँ पे हम बात कर रहे हैं ईस्ट की, जहाँ फेवरेबल कंडीशंस हैं, ईस्ट फेवरेबल कंडीशंस में ही रिप्रोड्यूस करता है. इट स्टार्ट्स टू प्रोड्यूस चेन ऑफ बर्ड्स और समटाइम्स सिंगल बर्ड्स. देन दिस बर्ड deattaches from the parent body. This is the parent body, and then this parent body and this bird deattach, and this starts to grow independent organism without any issue. Okay, we have also seen there has been growth of independent organism. Some cases only one bud has been formed, so only one individual will grow. In this case, several buds are formed, so multiple organisms will be formed after the detachment of the buds. Then comes spore formation. Spore formation, the example is fungus. Basically, you can take an example of bread mold. In this case, this dough in starting it looks like multiple fission only, but don't get confused with multiple fission. I will tell you why. See, in case of fungus, they have a filament. Fungus has a filament. And this is sporangium. This is sporangium, and inside the sporangium many spores will be formed. A large number of spores will be formed, and after the formation of these spores, the sporangium will burst, causing release of these spores over the surface, and each spore will grow into a new filament and then a sporangium. This spore will grow into a filament and form a sporangium, and each of them will do the same. But mark the difference I am telling you. In the case of multiple fission, nucleus was dividing inside the crust, but here it is not the case. No nucleus has divided. This is formation of spore. Spore is completely a different thing. No nucleus, no cytoplasm has been divided in case of spores. This is the spore. No division of nucleus or the cytoplasm taken place in the spore. Spore is completely different from those. In multiple fission, different cells were being formed, but here spores are being formed. That took place in unfavorable condition. This will take place in favorable condition. An example of bread mold can be seen over the piece of bread if it is kept for a few days, maybe 24 or 48 hours, and if it gets moisture, bread mold grows very fast. So this was all about the topic. We can discuss the doubts over our group. Okay, thank you.